Hello, everyone, and good afternoon. Welcome to our second community of interest, community of practice for optical genome mapping, a joint community presentation by the Laboratory of Medicine and Pathobiology program at the University of Toronto and the Canadian College of Medical Genetics. I think we've got a good number of people already logged on. We've got over 60 people logged on. So it's great to see everybody here today. We have a couple of presentations lined up for this afternoon. The first one that we're going to have, I'm very excited about, is Jean McGowan-Jordan, who is on the ISCN committee. The ISCN, very near and dear to our hearts, of course, the cytogeneticists. And she's going to be talking about the ISCN for OGM, which I think is something that's very important, and I'll let her elaborate on that more. But one brief announcement that I also wanted to make just to everybody before we get the program rolling officially was also a lot of people have asked me about the framework paper that's coming out from the International Consortium, and I just wanted to let everybody know that it is accepted at American Journal of Hematology and in press now. So we should have a footprint or something online, I would imagine, within about a week. So that is finally coming out. So I hope that that's good news for everybody as well. So I won't talk too much, and I'm going to pass things over to Jean and let her talk. And I think Jean also has some news about the ISCN, so she can share that with you, and we'll go from there. Jean?
Thanks, Adam. I'll share my screen. Thank you so much for the invitation to talk to you today, and for all those attending. Should be able to see my screen now. So this afternoon, I'm going to talk to you a little bit about ISCN 2024, which will include genome mapping nomenclature. I've been involved, as many of you know, with the ISCN Standing Committee for quite a number of years now. Currently, I like to think of myself as an advisor, but they keep giving me things to do. So kind of just the same as being a member. Anyway, so without further ado, I will try to make there we go. So just a little bit about process. We have the ISCN 2020, which of course didn't include any genome mapping nomenclature. This is the lovely group of people that I get to work with and got to meet with this past June in Glasgow. And we have been working on improvements to ISCN, as there always are things that can be made better. Hopefully, we've been working pretty hard actually, and we're hoping that the ISCN 2024 will come out sort of early or sort of springtime of 2024. There's lots of hard work, more frequent virtual meetings, and lots of email correspondence, all working towards that new and improved version of ISCN. And it's going to actually be quite a revamp in terms of restructuring. So a lot of focus on making things more consistent between technologies, and trying to put all the basic rules in one place so that it's a little easier for the user to find. A little more background. Since 2024, the ISCN has come under the Hugo Nomenclature Standards Committee, trying to work hand in hand with HGVS, as we have been for some time, for the betterment of the genetics community at large. So as you know, the ISCN has rules for karyotyping, for FISH, for region-specific assays of many different kinds, for microarray. Now optical genome mapping has been developed, and also for sequence-based nomenclature for structural rearrangements and copy number. Large I should say large structural rearrangements. As I mentioned a couple minutes ago, a big focus of ISCN 2024 will be improved consistency between techniques. So just as an example, if you look at this table, if you have an abnormality such as a deletion within band Q2 of a chromosome 2, by G-banding, we would designate that by showing the band twice, the same we would do it in fish. But for region-specific assays and for microarray, we just mentioned the band that is affected. I'll show you in a few minutes the new optical genome mapping nomenclature that's been developed, and tell you a little bit more about that progress. And it has options to report like we do because of the strength of OGM and the ability to characterize large structural rearrangements like you do with G-banding. It was felt that there needed to be a karyotype-like option, as well as more like a copy number or array-like option. So there are two different options you'll see in the examples that I give during the rest of my talk for many different types of abnormalities. Oh, I should say that I've kind of grayed out this sequencing part because that, although it's going to be revised in 2024, is still very much a work in progress. So I don't think the final decisions have been made there. There still needs to be more work done in conjunction with our HGVS partners. So just to give you some background on how we go about developing a nomenclature for a new technology like genome mapping. This initiative, although there had been some examples in the literature prior to this date, that when people were publishing OGM data, they would give kind of a pseudo ISCN nomenclature. The real effort started in September of 2021. At that time, we had about 3 examples. And then there was a reaching out to the OGM community. And so by the time August 2023 rolled around, we had about 76 examples of various complexity and type. You know, we really relied a lot on the community in this effort, which made it quite a bit different than the development of some of the other types of nomenclature. And so, you know, really need to shout out to the genome mapping community to thank them for all of that effort and collaboration. It has resulted in a publication that will come out in advance of the publication of ISCN 2024. It's actually just been accepted finally for publication in cytogenetic genome research this week. And you'll see the lead author is Dr. Sarah Moore, who is from Adelaide, Australia. She's a member of the ISCN Standing Committee. She's actually co-chair along with Roz Hastings, who's from the UK. And Adam and I have both been involved in the development of this new genome mapping nomenclature. So it's really been a very collaborative effort. A little long in coming. These things always seem to take a lot longer than you think that they will, but hopefully it'll be worth the effort. And I'll give you some examples now. So just some key points to keep in mind about the OGM nomenclature. If you really think about like normal results and like whole chromosome aneuploidy, OGM nomenclature is going to be very much like your region-specific assay and array nomenclature. So, and also keep in mind that there's not a specific nomenclature for neoplastic samples, unlike, say, G-banding. So the normal and abnormal OGM nomenclature is the same whether you're looking at constitutional results or neoplastic samples. Those of you who do microarray will recognize this sort of pattern of nomenclature where we use OGM as our little abbreviation, and then we for female we have X, and then the chromosomes 1 to 22 times 2. So very analogous to microarray nomenclature. Similarly for the male, or when there's the option when the sex isn't to be disclosed for some reason, to just leave those sex chromosomes out and just say 1 to 22 times 2. And you'll notice that there's no nucleotides obviously listed for these whole chromosome aneuploidies. So like microarray, when you don't list nucleotides, you don't need to mention the genome build. So it's just OGM and then your result. For whole chromosome aneuploidies, you have this example here, where if you have an extra copy of the Y chromosome, and then trisomies for, sorry, should have said, yeah, trisomy 8 and trisomy 21. In this particular example, they're just enumerated like that. So you're only listing the abnormal chromosomes, and you're listing the normal X here just for clarity purposes because of the sex chromosome aneuploidy. Okay, I thought I would just outline a few other clinical key points that, you know, a lot of them are very similar to other parts of the ISCN. So especially for microarray and RSA, like I said. You'll only be listing the clinically relevant abnormal results, although, as I mentioned, normal sex chromosomes might, or abnormal sex chromosomes might be reported for the purpose of sex determination. If you have multiple abnormalities, they can be either listed on separate lines or could be presented in a table. Aberrations are listed in numerical order from the lowest to the highest chromosome number, with the sex chromosome abnormalities always listed first, and that's uniform across technologies. Band designations are listed from Peter to Cuter. Multiple structural changes for homologous chromosomes are presented with the abnormality closest to the Peter described first, so the lowest nucleotide number listed first. And then just keep in mind that the nucleotides that are listed aren't necessarily those of the actual breakpoint, but are really to indicate the extent of the fragment that's determined by the position of the labeled DNA motif that's within that variant region. Okay, so here are some specific examples. I thought I'd start off first by talking about deletion. So I should also take this opportunity to say that Dr. Russ Hastings had prepared the earlier version of these slides. So I want to take this opportunity to acknowledge that I am reusing some of her slides because they had nice figures and outlined the key points. Okay, so here we have an interstitial deletion within band Q11.2. So as I mentioned earlier, you have the option, and because there are nucleotides, we have to specify the genome build, but you have the option to describe it either in a microarray, what I think of as a microarray type, copy number, nomenclature string, very similar, almost identical to microarray, or in more of a karyotype like nomenclature, where you specify it's a deleted 17 with these breakpoints. Like we do in G-bandy, you repeat the breakpoint if it occurs within the same chromosome band, and then you specify the nucleotides. You don't have to put ×1 because you've already called it a deletion. So those are the two different main options, here and here. And then, like we do for microarray, you do have the option to specify the normal nucleotides in more of a detailed format. So you can specify where your normal copy number ends and then begins again. So that's just more of a detailed form of saying the same thing as this top line. Just to note that just for readability, demarcating commas aren't used. They're optional in any case, but they can be used, except if you're using this detailed description. It really doesn't work because there's other commas in there and it will really mess you up. So they're not used there. There is the capability of indicating the proportion of cells with a copy number variant that can be estimated from the fractional copy number, and that can be included in brackets following the copy number. Likewise, if you have a constitutional mosaic, you can indicate that. For instance, in this case, it's an neoplasia, so you'd use the C designation as we would for other types of nomenclature. Okay, just thought I'd show you deletions involving the sex chromosomes. Kind of like I alluded to earlier, where for clarity, you're indicating the normal sex chromosome, even though we usually only describe abnormalities, but this is a deletion in a male of the X chromosome portion of the Q arm. So that's times 0, but we're just designating Y times 1 for clarity. Again, you have the option to designate this as a deleted X using the same nucleotide bands, more of a karyotype-like description. Inheritance is described the same way, where we have the MAT, PAT, DMAT, DPAT, INH, all of those abbreviations that are used just the way we do for other abnormalities or other types of technology. Okay, here's an example of a duplication. So this is a duplication involving band 6q21 to 6q25.1, and the nucleotides are specified using the kind of array type nomenclature. Similar to the deletion, you can designate this as a duplication if you have enough information that you're able to get out of your OGM data. So you can call it a duplication. Implicit in this is the orientation of that duplicated material. But if you want to make it even more explicit, you can use the detailed format to describe your duplicated 6 in this particular example. And in that way, your the orientation is very clear, because you're basically, like the detailed format does for other types of nomenclature, you're basically drawing out the chromosome from P-ter to Q-ter. Now, of course, if you have an inverted duplication, it gets more complicated, but it's the same principle. You can draw out the chromosome, the duplicated 6 chromosome, by using this detailed format. And in that way, make it clear that the material has been duplicated, but it's in an inverted orientation relative P-ter to Q-ter. Here's an example of an intrachromosomal insertion. So in this example, a small portion of the short arm of chromosome 2 is inserted back up into, as in described or sort of depicted in this ideogram, is inverted into band P24.3. In this type of abnormality, there's no copy number change. So we don't have a array type nomenclature. It really is based on how we would describe a similar type of abnormality by G banding. But because we're giving the nucleotides, we of course use the genome build, and then we outline the nucleotides in the second set of brackets, or the last set of brackets, let's say. You'll notice here the use of the tilde, and that's to describe any region of uncertainty. So probably use it a lot in OGM, I would imagine. So we've got our insertional, the place where the insertion happens is described ahead of the semicolon, and then the inserted material is designated by these nucleotide coordinates. Similar to the previous example where we have a detailed option, you can describe this intrachromosomal insertion as a derivative to chromosome, and then just basically describe it from P-ter to Q-ter with the join and reunion points designated by the double colons. And then similarly describe it in its nucleotides in the same way. Here are some examples of interchromosomal insertions. In the first one, the inserted material isn't identified, so that's why the query is used. But in the second one, this is a balanced 213 insertion where we have material from chromosome 13 inserted into band Q25 of chromosome 2. Here is the insertional breakpoint, and here is the material from chromosome 13. You'll notice that the nucleotides are reversed in order because the material that's been inserted into chromosome 2 relative to P-ter to Q-ter is inverted in orientation. Here's a relatively simple 6-7 translocation where we have, again, there's no loss of material here, so there's no option to use the array type nomenclature. You use the karyotype nomenclature to describe this balanced structural rearrangement. Here the breakpoints are both uncertain, so the tilde is used between the nucleotides, and all of this, these breakpoints reside within band Q21, so that's why there's no query here, because the band designation is not uncertain. Now, one of the nice things about the nomenclature is that if you have genes that are participating in, like to make fusion genes, they can be included in the nomenclature. They're not italicized, just like in our fish nomenclature, we don't italicize the name of genes. But they can be included in the nomenclature, and the fusion, of course, is indicated, as is common for all the Hugo nomenclature, by the double colon. So in this example, we have MECOM fused to ETB6 in this 312 translocation. The chromosome breakpoints are given here, and then the nucleotides involved in this rearrangement are designated in that next set of brackets. If you have a three-way translocation, just like in G banding, you would designate it starting with the smallest chromosome. So you'd have chromosome 6, then translocating material to chromosome 12, which translocates material to chromosome 21, and then it's understood that that goes back to the chromosome 21 material goes back to chromosome 6. These are the band designations, and then the nucleotide designations follow the string. This particular rearrangement results in an ETV6 RUNX1 variant translocation. So that fusion of ETV6 to RUNX1, because it's clinically significant, can be designated in that last set of brackets. Okay, now there are some complicated things that we need to be able to accommodate that are not always clear by the OGM data. So those of you who do OGM can attest to this, that a translocation between homologs may look very much like an inversion within that chromosome region. So if you have this instance, there is the option to use specific nomenclature when you can't tell, just saying that 16 is fused to 16. These are the fusion breakpoints. These are the nucleotides that are fused. And then we have fusion of CBF beta to MYH11. So that's specific nomenclature for these particular situations. For ring chromosomes, when you have basically loss of the chromosome ends, as is indicated in the Circos plot of chromosome 18, and you have fusion of a P-arm region to a Q-arm region. In this particular instance, the band, the band designation of the first breakpoint is uncertain. I indicate that in the nucleotides here with the tilde, but this span of uncertain nucleotides is such a big region that it includes several cytobands. So you would indicate that by designating the two extremities of the uncertain region with the tilde in between, and then the Q-arm breakpoint is designated. And the uncertainty of the nucleotides in the Q-arm breakpoint is again identified here by the tilde. And we have double colons on each side of that, of those nucleotides, indicating that the extremities are joined in a ring formation. This is a simpler example down here of a ring chromosome 8 with certain breakpoints in the P-arm and in the Q-arm, and those are the nucleotides involved. Oh, I think that's all I have. I will be happy to try to answer some questions if anybody has any. Anybody have any observations? Be kind with your criticism. The ISCN committee and the people who in the OGM community who worked on this are just people. And we tried to do our best and think of all of the different scenarios that we will be faced with. I'm going to stop sharing my screen in case it makes it easier to ask questions.
Thanks, Jean. That's great. There's one question in the chat from Laura, and she's asking, how do you know it's a ring in OGM? You only see a deletion on the P-arm and the Q-arm.
You should be able to see the fusion breakpoint with your molecules spanning the two breakpoints or across the breakpoint to see that joining of the P-arm to the Q-arm.
Yeah. Kathy?
Hi, Jean.
Hi,
just
Kathy.
wanted to say, how are you?
Wow.
Great presentation. Thanks so much. And also thank you for all the years that you've devoted to the ISCN committee. I know it's been a ton of work, but we really appreciate your efforts. I just had a question about the direction. Because, you know, cytogenetically, we've always used the centromere, like relative to the centromere, right? So, and unfortunately, some of the examples in ISCN have always been on the Q arm rather than the P arm. But okay, so let's pretend there's like an insertion, an intrachromosomal insertion from the Q arm to the P arm of a chromosome, whereas by cytogenetics, traditionally, we would have probably said that a direct insertion would have been relative to the centromere, right? So if it went into the centromere, it's actually being inverted, right?
Yes.
But it's actually direct cytogenetically.
Well,
before.
we actually got away from using that terminology. You may have noticed it was subtle, but deliberate. And that is because we really need to think about, regardless of the technology, think of everything as if you know the nucleotides, even if you don't, like say you're using G banding. You need to, we need to change our
way of thinking.
Yes, essentially, to always think relative P-ter to Q-ter. So yeah, we got away from using those direct and inverted insertion type of phraseology.
So if that, so say the Q, say it's, I'm just making this totally up because I don't know what chromosome I'm talking about. Q11.2 is close to the centromere, and then say the distal breakpoint is Q35. if that's going to be inserted into the P arm, you're going to put it, if it's supposedly direct, it's going to be Q11.2 at the top and then going to Q35, right?
I think so, if I can imagine what you're trying to
Yeah, so
describe.
it's,
I
you're just
do better with
moving it.
pictures.
I
Yeah,
know. You're just moving it from the Q arm to the top arm without changing
yeah,
the orientation, and that's
exactly.
basically considered a direct.
That's right.
Yeah. Whereas if you actually turned it around, it's inverted, whereas traditionally it would have actually been direct. But anyways, okay, yeah, I just want to clarify that it's always, you're just going to think of any movement as direct if you go P to a P-ter to Q-ter.
Yes.
Yeah. Okay, great. Thank you.
Thanks, Kathy. I see there's another question from Melanie.
Yeah, so maybe I'm having a hard time with my brain, but how would you be able to distinguish by OGM an insertion, for example, of 2q36 into the P arm of the same homolog chromosome 2 versus an insertion into the other homolog? Because by OGM, you're not able to distinguish between, unless the breakpoints are so close to one another that you can actually have molecules that span the whole thing, you would never be able to distinguish between the two, right?
I agree. And like many things we do in cytogenetics, it will be an interpretation of the data.
Okay.
So you have, you'll see it as a deletion in one arm, for instance, and then you'll see the breakpoints spanning that region that's missing in the Q arm, for instance. In the same, the breakpoints would be in the same molecule, so you'll see that join and reunion within the P arm. Oh, you're talking about you're talking about an insertion, right?
Yes, but I mean, you would see you would see the joining of the Q arm segment into the P arm. You would see that structural variant, but you would not be able to confirm that it occurred on the same homolog.
Nope.
It could have been on the other homolog.
It will be an interpretation of your data.
Okay, so unless you can
G-banding,
confirm,
Melanie.
Yeah, well, that's the thing. I'm
You
thinking,
can't
like,
get rid of
unless you can.
G-banding yet.
I know, I'm happy about it, but it just means, like, so when so it's something to consider if you're going with the karyotype like ISCN, you have to be pretty certain of. I mean, sometimes it's really obvious, but sometimes when there's uncertainty, you might be better off with the array-like nomenclature.
You might be, but in that case, there would be no copy number change. So you have.
Oh, you can't. You have to use a carry.
Yeah.
So you're kind of. It's a or. Yeah. Oh, okay.
There's a question in the chat. Adam, do you mind if I?
Yep. Yeah, go ahead, Gina. I was going to get to it, but go.
Eric McGinnis asked, sorry if I missed it in the presentation, for the CNV FCN, you can be used to report the proportion of the cells within the abnormality. For structural variants, is the ISCN recommendation to include the approximate proportion? I believe we're recommending that just be included in any descriptive text, not in the actual ISCN string. If I recall the details of the manuscript.
I think, but Gina, I think that proportion is, so you have to convert both FCN and SVVAF to proportion.
Yeah.
I guess depending on the sample. So if it's, especially if it's somatic and you wanna sort of have an idea of what the relative levels of each abnormality are. So what we said in the manuscript was that each of the abnormalities has to be converted appropriately to their proportion of cells.
Right.
Yeah.
Right. Okay.
So, and the calculation, depending on whether it's, whether it's an SV with a VAF or whether it's a copy number abnormality, is a little bit different depending on. I have a table I can show later that.
Oh, that'd be great.
Yeah, that kind of summarizes So most of it's clear, and then it gets into some stuff that gets muddy. Like when you get amplifications, what's the proportion of cells that have an amplification?
Yeah, that's going to
And,
be tricky.
yeah,
Very
that's
tricky.
when I hide under my desk and say, "Don't call me."
Yeah.
So yeah. Yeah, most of the time it works. And in fact,
It's-
even BioNano said to me, like, oh yeah, they said anything over a copy number of 4 breaks the algorithm. So for the automatic VAF calculations or whatever.
Good to know.
Yeah. There's another question from Susan. She says she's curious if we can provide an example of complex rearrangement and the nomenclature. Say for those cases that have multiple rearrangements involving multiple points within a chromosome, or a couple or a few chromosomes with OGM.
I don't think it made it into the manuscript, but in the chapter that will come out in ISCN 2024, there will definitely be some complex rearrangements that have been worked through and reviewed by numerous people numerous times. So yes, I don't have that at hand today, but they will be forthcoming for sure.
Are there any additional questions?
So we hope that this manuscript is going to be published before the end of December, because it was just accepted this week. So hopefully, fingers crossed.
It's really important, I think, for the community moving forward to have that
Absolutely,
published.
and
yeah,
I do feel kind of badly that it's taken so long. It was just, it was a big undertaking, and important to get input from all of the stakeholders that did contribute. It was really a group effort.
fantastic. Thanks so much, Jean.
You're welcome. Thanks everybody for your attention.
I'm going to, since we're on the topic, I'm just going to walk through a couple of slides about fractional copy number considerations and VAF for everybody. So just to sort of, this is more my opinion at this point. So I invite, this is why we're here in this community. we're inviting comment and discussion in case anybody disagrees, because I think this is also something that it's great to have consensus around how we're going to report this stuff. So just to sort of point out that the way we look at this data in the different modalities that we use is slightly different. So on the left hand side here, you can see like these are the karyotype in fish or cell level modalities where you're actually looking at abnormalities within a cell. So you're saying, oh, there's a certain proportion of the cells that have that abnormality. And so, both for karyotype and for fish, this is the same kind of idea. So, I mean, if you were living in a perfect world where you actually got this level of concordance between karyotyping and fish, well, that would be great. But when we move to any, and this is not an OGM specific problem, this is any bulk DNA assay, the minute you move to microarray, MLPA, sequencing, whatever you're going to do, you're then starting to look at the result based on the presence of the number of copies or alleles, not in the cell level, but just basically in the proportion that it is in the sample. So in order to sort of make all of these numbers and the structural variant frequencies all make sense and be interpretable together, the ISCN has opted for converting everything to a value that is proportion of the sample. And so, you have to do a little bit of mathematics to sort of figure out some of these things out. And so, here's sort of our standard case in the middle, where you've got a copy number or fractional copy number of 2, and then you would say this is your normal diploid state. And of course, you wouldn't report any abnormality in terms of proportion of the sample. So when you lose copy, you have some level of mosaic monosomy for, so let's say in the context where you had lost a whole chromosome here, and then you can see here your fractional copy number represents a certain percent or proportion of the sample. And so this is how that translates. Now it gets tricky when you cross thresholds from, say, your for your monosomies down to your nullosomies, where you get combinations of those. So when you cross from monosomy to nullosomy, it doesn't go evenly, right? It doesn't jump evenly to, back to sort of 10% or whatever. You have to make this jump up to 55 through to 100. So you can see that the actual proportion, the calculation is changed. Now we are making a couple of assumptions here. So when we're looking at this and we're saying, okay, this is a mosaic monosomy, and then once you move below 1, this is a mosaic nullosomy. And it could actually be a combination of nullosomy with monosomy. And so this is sort of one of the caveats of this kind of reporting, is that we're not actually pulling out that sort of that cell level resolution. That cell level resolution's being lost. And so we're just sort of saying that, the fractional copy number is, of whatever this means is present in that percentage of the sample. And it means the same thing going in the other direction. So again, as you're moving up from 2.1 to 2.9, you move up through 10 to 90% mosaic trisomy, then trisomy, and then up into tetrasomy and beyond potentially. So I made up this chart just to sort of like make it easier to report proportion of the sample. And of course, the caveats being that, like sometimes you can have, a clone with +8 and a clone with +8, right? So you've got trisomy 8 and tetrasomy 8. And so conceivably that's going to fall somewhere in here, but you're not actually going to be able to separate that information out using OGM alone. So again, these were just some general questions, to make to basically illustrate those points, to make people think about, like what happens when, your fractional copy number is 2.5, and that's, trisomy 8, what proportion. So you can go back here and you can sort of read, okay, 50%. But then, the other problem is, of course, when you move on to the sex chromosomes, which are normally present in only one copy, then you can't use this table. You have to use a different table, right? So, because you're starting from one chromosome. And in fact, when you only have one chromosome, it's a lot easier because you don't have to do any complicated math. If it's 0.9, it's 10%, it's really easy. So, anyways, the fractional copy number for the sex chromosomes in general, when there aren't multiple copies of them for whatever strange reason, this obviously gets a lot easier to report. So this is sort of what that conversion looks like. And so converting VAF is also relatively straightforward, but there are a couple of things that are interesting to consider. So VAF, usually for OGM, we'd only be reporting down to 0.05, but essentially anything between 0.01 and 0.5, you just basically multiply this to get your proportion of the sample. So if your VAF is 0.1, it's in 20% of the sample. If your VAF is 0.25, it's in 50%. And if your VAF is 0.5, it's in 100%. So again, that's a fairly simple calculation. But what happens when you have a VAF that's 0.65? What proportion of the sample is it? You can't multiply it by 2. It's not 130% of the sample, right? So when your VAF is over 0.5, my personal feeling is we're just reporting the VAF as it is. If it's 0.65, then we report 0.65. And what are some of the reasons that you can have a VAF that's over 0.5? Oops. So you can have a lot of, you can have things where you have a deletion on one chromosome and you have an SV on the other in the same region. And so you've reduced that particular region to hemizygous. And so you've now actually got 65% of the remaining chromosome that's affected by that rearrangement. You can have things like loss of heterozygosity that results in an abnormality actually being present on both homologs at the same time via recombination event. You can have things like changes in ploidy. So you could have, for example, triploidy for the whole genome, which would give you 33% percentages for each of the remaining chromosomes. So if you had the abnormality on 2 chromosomes, you'd have it in 66% approximately, if you had 100% cancer cell fraction, for example. So there are lots of reasons where you can get a VAF of over 0.5. And so I think those are just things that need to be carefully considered when any interpretation is written they're sort of well considered. Those possibilities are well considered when you're writing about those VAFs. So anyways, that's basically what I wanted to say about the VAFs. And just to add, Tracy Tucker had asked me a question too about cross-validating abnormalities that are seen, say, by karyotype, and then something slightly different seen by OGM. And I'm not sure that that I have a perfect answer to her question. But what I did want to show in that regard was that we are, well, when I say we, I mean we in my past life, we've done some work where we'd looked at a variety of abnormalities, using karyotype fish and OGM. And I think the overwhelming take-home message is that we actually saw relatively high concordance for these abnormalities across modalities. And there are some exceptions here. So for example, there was one sample where we saw a del7q by karyotype with not seen by fish, not seen by OGM. And so what's the possible interpretation here? Is it a karyotype false positive? Is it metaphase bias? Is it the cutoff? And by metaphase bias, I mean that the cutoff for fish is too high to detect the abnormal clone, and the same being for OGM, the LLOD is just not there. So you're seeing a slightly higher proportion by karyotype than you would by an interphase modality or a bulk genome test. And therefore you get this kind of discrepancy. So what do you do in these situations, when you're doing your validation? And, for the most part, I think overall, we have to take the hit and sort of put a little star up in the corner and say, there's a couple of possible interpretations here. So, you're not, in these cases, the question is, also, is this real? I mean, you can't confirm it with two other modalities. Jean?
Did you try sequential fish?
G to fish? No, not for very many cases. We didn't. I mean, we've done that in cases, Jean, where we've had like G-band positive del20Qs and then interface fish negatives, and then we've G to fish them, and we've been able to show that they are real. And then the question I have is, okay, so they're real, they're in the metaphases, but in what proportion of the total tumor are they? If they're so small, we can't even detect them by interface fish. So, then we get into sort of questions about clinical relevance, but yeah. So, I mean, in a lot of cases, they can be real, but just very low level. Or actually, we had a really, really interesting case. I give this example all the time. So if you've heard it before, just please ignore me now. But we had this case where we had a patient with CML come in, and we did the karyotype on the patient, and the patient had, I think it was over 50% of metaphases with tetraploidy. So they were 92 with two Philadelphia chromosomes because the whole genome was duplicated in a large proportion of the metaphases. And the clinician called me all freaked out, and he's like, "Is this blast phase? There's like extra derivative 20, And I was like, okay, let's calm down, and let's see what the proportion is by fish." So we fished it, and we didn't see a single a single interphase nuclei with tetraploidy by fish. So there's clearly a gap between the metaphase and the interphase nuclei, and I don't think there's any good. I mean, what's the biology there that says why a particular karyotype might have an advantage, and therefore we're seeing it at a lot higher proportion than you are in interphase nuclei? Or is it just simply you're diluting out? Because you're looking only at metaphases, you're diluting out all the other blood cells, and so the proportions change significantly. And I don't think that happens very often, that we see those kinds of really large changes, but it's difficult to say. No, Susan put in the chat different cultures. It was the same culture actually. So yeah, it was a bit of a mystery. So I mean, these are things that can happen. And I think they're just challenging. They're just challenging to interpret. But just throwing that out there for food for thought for the interphase metaphase bias. Always keep that in the back of your mind when you're thinking about validating these things. So another example here was a Del 20Q seen by karyotype, seen by fish. We had actually an SV call at the right breakpoints, but no copy number call by OGM. And so again, this is one of those questions where it was just probably above the LLOD for the SV call, but below the LLOD for the CNV call. So it was probably in this ballpark in terms of its actual percentage proportion of the cells, right? So sort of 15, yeah, exactly, 15% or so. So these are things that you're going to see anytime you have technologies that have slightly different limits on their ability to detect things. But so it's something to consider. I think the larger point that I wanted to make, though, was just looking generally at the data. It's very consistent across the modalities for the most part. We don't expect it to be the same. We expect there to be some difference in terms of the percentages for some of these things. So very similar for aneuploidy. Again, we had a case here that had a plus 14 and 9% by karyotype, not seen at all by fish or by OGM. So again, what does that mean? So the same kinds of questions: false positive, metaphase bias, et cetera, abnormalities, things of +8. This again, just sort of slightly different numbers here, but very, very close. And again, remember the sampling percentages are a little bit different. So karyotype usually 20 metaphases versus 200 nuclei versus 300-400x coverage. So slightly different statistical depths here. So we do expect to see some differences. This, for example, was a false negative by karyotype, so the monosomy 7 wasn't seen. And just some differences in terms of sampling we're seeing. But overall, the great majority of samples actually had fairly consistent results. And then this is the same for a whole bunch of structural variants. So again, you can see, basically actually here much more consistent overall in terms of the concordance between techniques. This, just a note here, this one wasn't actually identified as a 12-19 by karyotype. I think it was an add 12, add 19. So that's one of the, we saw the 12 and 19 and that roughly that percentage. So again, it was close, but and then the other, I think the interesting one down here is this T411, only seen at 8% by karyotype, and then very, very high by fish and by OGM. And we actually think that the 411 was cryptic by karyotyping, that there was a clone that had like an add4Q, but the 11 looked relatively normal. So there was probably something going on there that wasn't sort of resolved by karyotyping, but was seen clearly by the other techniques. So let's see, were there any comments there in the chat? Let's see. Sometimes mosaic, Melanie writes, sometimes mosaic aneuploidy is not called by the algorithm, but when you look at the whole genome, yes, exactly. So, that is one of the things that we recommend. It's also recommended in the framework paper that when you look at the whole genome view, when you're doing an interpretation, if you can see any deviations in the copy number that you can clearly visualize that aren't called with the recommended filter settings, you can sort of take the filter settings and you can move them to all, which will then give you a little bit more of a messy result, but it will potentially call those regions. So if they're really distinct copy number changes that aren't called on the recommended, and the recommended settings are quite stringent for copy number, you can definitely move to all and then make a judgment call in order to make a copy number call for those. Okay, so that was all I wanted to say about that. I don't know if there's any other questions or comments about that.
I just had a quick question. Somehow I have it in my head that we don't always get a VAF value, I guess it depends on the algorithm, but if you don't have a VAF, do you always at least have a fractional copy number?
Yeah, so
Is
if
that what
you
it is?
if you have a if it's a copy number, if it's called by the copy number algorithm, you'll have an FCN value. If it's called by the SV algorithm, you'll have a VAF. Except depending on which version of Solve you use, I believe if you go back, if you have cases that you've run previously in Solve 3.6, I don't believe Solve 3.6 was using VAF. That was something that came in 3.7 and later, so that you that may actually be the issue, Melanie. If you don't have a VAF, it might be an old version of Solve. So I don't know. Eric also has a comment.
Great, thanks, Adam. That was a really good discussion of the copy number problem we've been thinking a lot about lately. So as you know, we just kind of went live with this recently, and the copy number problem of what to report has been on everyone's minds. So just to clarify, because I saw in some of the slides that there is this placement of square brackets after the variant call in the ISCN nomenclature indicating proportion of cells.
Yep.
But then I've also heard recommendation is put in the text. So is the suggestion in the ISCN string or table or whatever you're doing, include your variant call and then in square brackets proportion of cells, or include nothing and simply describe that in the text?
We've been including proportion of cells in square brackets in the ISCN string or in table format, and then of course also in the text interpretation as well. That's the way I've been doing it. I find that when you get into the more complex samples, like whenever we get into an ISCN string that has more than about 3 items in it, we basically have been defaulting to table format because it's just a lot clean. I just find it's a lot cleaner to report. So I think that might be helpful. We do have report templates as well that are in the framework paper and the supplemental methods. If so, you can, and I believe there aren't report templates, but in the ISCN manuscript, there's also a section that outlines a couple of different like table formats that can be used.
Great, thank you. Yeah, I'm not sure if our LIS allows us to table, but we had one with 4 abnormalities and it was getting quite lengthy. So the line by line might be a good option. I just wanted to echo your point about the metaphase bias. We see that a lot with CLL, actually, not metaphase bias, but culture bias, where trisomy 12s and del13Qs show up in culture, but not in our OGM specimens. That's been posing a problem for validation of those types of specimens. Yeah.
We had a patient with dual diagnosis, AML and CLL, and we actually had a blood sample and a marrow sample in the patient. And you could see all the CLL abnormalities in the blood sample and all the AML abnormalities in the marrow, and not vice versa. So yes, there's certainly the compartmentalization of the cells of interest in certain diseases is an issue.
Thank you.
Hi, Adam. It's Nassim.
Hi, Nassim.
Yeah, I wanted to ask you a question about these coordinates in the nomenclature. So sometimes the abnormality is called both by CNV and SV algorithms. Yes. And you know, these coordinates are different. So if we want to add these coordinates, which one we should use?
Yeah, I mean, so when you have the option of both, my preference has been to report the SV. My preference has been to report the SV rather and not report the CNV data. And the reason for that is that the SV data is based on labels, right? Whereas the CNV data is actually based on a sliding window type call, like very similar to a microarray type. Where it looks at the coverage of the molecules in that region, and then it makes a copy number estimate, and then so it's less precise. And I think that's Melanie just said basically the same thing in the chat. So she said, we've been told by BioNano that the SV coordinates are more precise than the CNV. And I would agree with that. And I think the other issue I think that's relevant is that, you know, that the advantage of the SV call is that it's a call that's in context. So obviously a deletion's a deletion, but, you know, I think the thing that's important is that, you know, when you have the SV information that puts whatever the copy number change in the context that it is, whether it's an unbalanced, say, translocation, or whether it's an interstitial deletion, if you actually have those, if you have that information from the SV algorithm, it's better to report that. It's a more complete interpretation.
Thank you. And have you tried VIA?
I have not tried the VIA that comes with 1.8, with Access 1.8. I tried a very early access version of VIA, so I don't actually have a lot of experience with VIA at this point. Yeah.
Okay, thank you.
Yeah, I think, well, I mean, 1.8 is sort of getting rolled out to most people, so we should, I mean, we should see it. I mean, again, I think VIA is supposed to help as a reporting tool, because it really helps to sort of put your put all the SVs you have, and you can apply rules and filters to them in a more, I don't know exactly what I want to say here, like I want to say organic maybe, but more, it's just easy to sort of apply a bunch of different rules without having to always reload the browser. So when you do this kind of stuff in Access, you're always waiting for the browser to reload. So I think the advantage of VIA there is that you can apply sort of filters or rules to the SV calls and sort the data without having to do this sort of browser refresh every time. So that's sort of helpful from a reporting perspective. That was great. I think, you know, I don't know what everybody thinks about. I actually think that maybe for our next, COP meeting, we should maybe have somebody give a presentation on VIA. So if there's anybody out there that wants to do that, you know, reach out and we can plan something. Okay, Tiffany has a question.
Sorry, this is Doha Itani from St. John Regional Hospital. We are logged in using our manager's account. So we have been using VIA post 1.8. The interface is much more friendly than Access. You can go through an entire case without getting frustrated. But the table format and the actual generation of the report needs a lot of work. So that it will generate fantastic, comprehensive results, but not necessarily clinician-friendly reports. So that needs to happen. When is our next meeting? Is it in a couple of months or next month?
Yeah, it's these meetings we're planning to have them quarterly. So
okay.
Yeah, there would be, the next one would probably be in around February or March.
Okay. By then we may be able to give a talk.
Okay. Well, we can chat offline.
We can chat offline. Yes, yeah, that's why I wanted. Okay.
Okay. Awesome. Thanks. Okay, great. At this point, if there's any other items for discussion that we want to talk about, we have some time for that. I know Zaid also has a case or a case or two he wanted to present, and time permitting, I also have something that I will present if there's time left and no other items for discussion. So at this point, I'm going to turn it over to Zaid.
Great, thanks,
Zaid,
Adam.
if you want to do both your cases, one right after the other.
Yeah, I'll be mindful of time. I think they shouldn't take too long. Just, I guess, pretty relevant to the topics you've already brought up, I think. So it'll be good.
Okay, awesome. Thanks very much.
Absolutely. So yeah, thanks for letting me talk a little bit today. For those of you that don't know me, I'm a lab scientist at Vancouver General. I work with Doctors Tara Spence and Eric McGinnis, both of whom are on this call, I believe. And we've been validating optical genome mapping for some time. As Eric said, we've finished that. And here were just two interesting cases from our validation cohort that I think touch on some pertinent topics. So the first case was really this one with complex karyotype. Most findings were unambiguously detected by OGM. That includes the +6, as well as the +8, the +19. These two interstitial deletions, del 11 and del 20. But the one of interest here, of course, is what was originally called as a del 18, turned out to just be more complex than we originally thought. And in fact, it was this T818 that resulted in a SND1 SETBP1 fusion. And fusion genes are, of course, recurrent molecular abnormalities in AML. They're some with significant diagnostic or therapeutic value, but in this case, SETBP1 mutations do have some prognostic significance. So here we identified it as well as a novel fusion partner that was originally missed by our standard of care methods. Going back, that really resolves, or I guess captures, both the findings that were originally detected by standard of care, but as well as just some novel additional findings. I'll just hop right into the second case. Oh, sorry, actually, I just wanted to touch on this. So the del 20 and the del 11 are these interstitial deletions. And what I really want to focus on is the fact that OGM uses two distinct pipelines or algorithms to make these calls. As we've already touched on today, there's the copy number calling algorithm that's really, as Adam said, looking at a molecule level, whereas the structural variant pipeline is looking at a label level. And obviously, looking at a label level, we have finer resolution, but really it's added support to have variant calls from both the structural variant pipeline as well as the copy number variant pipeline when it comes to detecting interstitial copy number losses. So in this case, there was a del 11 that was called as an intrachromosomal fusion 1111, as well as this copy number change spanning the exact distance. And so looking at on access, you can see a consensus map that is half spanning to one side and half spanning to the other side of this massive copy number loss call. And so this would be the copy number call correlating with this ISCN, and this would be the structural variant call, or the intrachromosomal fusion that is shown here in the circles plot. And there was another case of this on the del 20. I forgot to update these pins, but this should be pointing over here to the del 20, which also similarly shows a intrachromosomal fusion call spanning the distance of this copy number variant. So this again is just added support. This has been of interest for us recently because we've seen these cases with relatively small but cytogenetically visible copy number losses that aren't associated with a structural variant call, but are interstitial. And so the added evidence needed for supporting this call as being real would typically be this intrachromosomal fusion call is what we've sort of concluded. And of course, the aneuploidy for chromosomes 6, 8, and 19 were detected, and that was, again, unambiguously evident from the copy number pipeline call, or the copy number pipeline. So I'll just jump right into the second case. The second case has interphase FISH done for BCL6 and MYC using break-apart probes, as well as an IGH BCL2 dual fusion probe set, which wasn't shown here. But the BCL6 probe set pattern was within normal limits, not shown. But the MYC probe shown here has an abnormal signal in about 68% of cells, by a split signal, evidence of some MYC rearrangement, which is usually with IGH. Now, the IGH BCL2 probe showed one fuse signal in 75% of cells, consistent with the presence of a clone having a balanced reciprocal translocation, a T14-18. These results showed the presence of two cytogenetic abnormalities. That's a MYC rearrangement and an IGH BCL2 rearrangement, which is in keeping with a double hit lymphoma in this case. But OGM was able to show a translocation between chromosome 14 and 18, and 8 and 12, which upon further inspection showed an IGH BCL2 rearrangement as expected between 14, 18, as well as a MYC LRMP rearrangement between 8 and 12. And so this really further refined the classification of this double hit MYC non-IGH rearranged lymphoma, which of course defines a prognostic category in WHO 5 and defines about a 25% difference in overall survivability between the double hit MYC IGH and the double hit MYC non-IGH rearranged lymphomas. So overall, this was just a case that showed sufficient evidence for high-grade B-cell lymphoma with MYC and BCL2 rearrangements, but the key takeaway was that OGM could distinguish in an unbiased manner what MYC was rearranged with without any prior knowledge needed for multiple FISH probes, for example, and to identify possible MYC fusion partners. So those were the two cases that I thought were of interest. Any questions?
Eric answered my one question in the chat, which was about whether this was a bone marrow or a tissue. I mean, this is one of the big things I think with the lymphoma is that how valuable OGM would potentially be with all the things we kind of have to do in lymphoma now. You know, with the sort of Burkitt-like 11Qs and all of the non-IG, like the IG partner being really important for MYC. It would be great to have an ability to get. I mean, we sort of thought the best way might be to do this via cytology to get cytological aspirations, but we only tried a couple. They did work, but we didn't do a lot of work on this. So I think it's you know, it's certainly a super interesting application for OGM. Mm-hmm. And then the only other comment I made was that we noticed that the CNV pipeline can make some wonky calls when there's a call that crosses a masked region. So because there's several regions in the genome that have CNV masking, and so then when the copy number caller goes over the masked region, sometimes it gets a little bit wonky. You see it on the genome plot when yeah, it's
maybe like well, this one is I'm pretty sure this one right here was an artifact and in somewhat proximity to the centromere.
You see that little blip that's just below the 1QH? You got a tiny little red blip there.
Yeah.
Yeah. So that one's coming right off the end of the 1QH. It's probably an artifact of coming off the mask region. Mm-hmm. So yeah. There's a ton of seg dupes in there too, so that doesn't help either.
For sure.
Yeah. Any other questions for Zaid? Ken writes, yeah, tissue samples for lymphomas on OGM, would fresh core biopsy that would typically be sent for a flow be a good starting material for extraction? I think so.
I'm not too sure.
I don't know. I think the big problem we always have is that we need the number of cells to start to process, right? So you need that 1.5 million cells. So I don't know if Anne, are you still on the call? I know Anne Tierens was on the call. Maybe she could comment on cellularity on those samples. Doha?
For tissue, there's a protocol with BioNano, and the consideration is frozen tissue, and there is a size of the piece of the tissue that you need to collect, and they take into consideration the cellularity of the program. So I did have another question. I'm sorry. So first, to summarize, I think in terms of tissue, the cell number is not going to be something that is totally required if you have the size of the tissue collected properly. When you, and now my question, when you collected the FNA samples, did you process them fresh or did you freeze them, you know?
For these
lymphoma samples for that, I think Adam tried that a couple of times, right?
Yeah, various people have tried it. Nikhil, who was at Augusta when he did a lot of his development, did a lot of FNAs. I believe Nikhil processed fresh. I can't remember. Our samples, I believe, were I think we stabilized them, and then they were 1 or 2 days in the fridge, and then they were processed directly. I don't know. Is Anna? I think Anna's, maybe Anna. Anna, you're on the call. I don't know if you remember. Remember that cytology specimen we got? I can't
Yes.
remember exactly how we processed it.
When we processed fresh,
Yeah.
well, fresh from it had been in the fridge in the flow lab, but when we received, we processed immediately. The other one, I believe it was stabilized and frozen. We collected just the cells, centrifugated, collected cells, and froze it. But I'm not absolutely sure on that. It was done by Olivia.
Okay.
Okay, thanks.
Nope.
I think the other consideration too is I know that there is coming a lower input protocol, so like a 500,000 cell input protocol. So I think for smaller samples that's going to open up the possibilities, I think, for these smaller types of biopsies, probably to be able to get them onto OGM. So I know there's a white paper coming, but it hasn't been released yet. So Ken makes a comment about 10 millimeters being difficult. But you know, if we had clinician buy-in, that we could maybe do that. I mean, this sounds like a perfect like QI project, you know, for somebody to do, you know, that to see if we could get better information and figure out a pathway to make it work.
Is it 10 millimeter by 10? I thought it was 10 millimeter by 1, or it's a certain grams, like you can weigh it. I don't remember the protocol. We've been collecting a few samples here and there and freezing them, just stabilize and freeze for now. And I believe we've been weighing our samples. They give a weight also. So it's not a big piece of, it's not like 1 by 1 cm. It's more like 1 cm by 0.2 or 0.3, something like that.
Okay.
Like a core.
Yeah.
Just to kind of add to that, to Tiffany's point, I think there is a milligram amount in their protocol. I think it's like 10 milligrams. And then you flash freeze it.
Yeah. Yeah. We've been weighing more than measuring for that reason.
Well,
yeah.
And so, and here's the consideration, right? Because I think the way the whole extraction protocol for BioNano works is it's all based on sort of having the right like stoichiometric proportions at each step. So if you look at actually what you throw away at each step, you throw away a lot of material, not throw away, but there's a lot of residual material that's kept at each part of the process, right? So you do a DNA extraction, and of that DNA extraction, you put like, I don't know, a tenth or something into the labeling reaction, because there's always material to go back, or most often there's material to go back to. And then you do your labeling and you load a third of that onto the flow cell, right? So there's always a lot of material that I think is left over. And so I think this is the whole point of the low input protocol, is that they have to figure out a way to put less in. So they have to adjust all the volumes, the downstream volumes, so that you can put less input cells, but still get the same result out. So anyways, I think it's going to be exciting when it finally comes out and we can try it, because there's just a lot of applications for the lower input. Well, okay. Zaid, again, thank you so much for bringing some cases to present. You know, this is the point of this group, is we want people to bring their cases and show them to the group. They're, you know, when they're interesting, when they're hard to figure out or hard to understand, or when they illustrate, you know, some kind of really interesting point. So, you know, again, for future meetings, please don't be shy. Please volunteer to present cases so that we can keep the momentum going.
No, absolutely. My pleasure. Thanks for having me, Adam.
Thanks so much, Zaid. I wanted to, I have one last presentation, which is mine, which was just basically a summary of a poster I presented at AMP 2 weeks ago. So if nobody minds, I'll just take 5 to 10 minutes and present that. I just wanted to, and then that would be it for today's meeting. That's all we have, unless there are any other points for discussion. So with that, so when I said a poster, I really meant it. no, just kidding. It actually is a PowerPoint slide, and we'll go into some more detail on some of the items. So what I really wanted to show, and I think probably maybe some of you have already seen this presentation before, but this is a summary of 55 acute myeloid leukemia cases that we had done in our validation for OGM, but that we also had molecular data for based on the our NGS myeloid panel. And so the idea, what we wanted really to do was do a couple of things. We wanted to sort of put all the data in this oncoplot style figure where we could look at all of the different cytogenetic categorizations from the ELN, and also all of the molecular categorizations, including CBP alpha and PM1 FLT3 and mutational status for other important genes. So we could get a nice sort of overview of what was going on. And then at the end of that, what we wanted to do was actually look at the ELN 2017 prognosis and the ELN 2022 prognosis, and actually look at, what actually all of this additional information did in terms of reclassification. So what the reclassification rate was, etc. So let me walk through just a couple of things really quickly. Oops, that would be the last slide, not the first slide. There we go. Okay. So you can see a lot of these blue boxes, and these blue boxes basically indicate where OGM detected something that we didn't detect with conventional testing. And so, a lot of this stuff isn't very surprising. We had several cryptic KMT2A rearrangements. We had several cryptic MECOM rearrangements. And we had, in addition, some cryptic NUP98 rearrangements that were detected only by OGM. And so obviously, these kinds of findings are not terribly surprising anymore, as there's been several publications out now indicating that this type of resolution is why we like this technology so much, and the fact that we can see these things that are, not impossible to detect using conventional techniques, but it is challenging, because I think, the alternative way to do this is, to have to run several ancillary assays, whether you're, running, copy number assays via array plus minus RNA fusion panels, or you're using a whole panel of FISH. So you're having to run a lot of additional assays to get this information. There was actually a recent publication from the Mayo Clinic where they compared their next gen sequencing methodology called MP-seq. The MP-seq. Anyways, it was something, they have some neat acronym for it, and it basically is able to capture a lot of these rearrangements and structural variation. And they compared it to conventional cytogenetics and to FISH. And, the conclusion of that paper was actually that a very good FISH panel, a large FISH panel, and karyotyping had about the same sensitivity overall as MP-seq did. However, I think that, the MP-seq also comes with some significant limitations, including its ability to actually get lower level copy number abnormalities because of the depth at which it's performed. So, this is again where OGM has an advantage over some of the sequencing-based techniques because of the depth. Anyways, moving on, what we really wanted to show was the difference in the reclassification. And this was something that, we have a reclassification rate in this very small cohort, relatively speaking, of 25%. But there, in fact, are some, I think I have, did I copy it? Yes, there are 2 papers out now in the literature. There's one from this group at Knight Cancer Institute in Portland, which is a comparison and validation of the 22 ELN guidelines on approximately 500 patient clinical trial set of acute myeloid leukemia, where they showed an overall reclassification rate of about 15%. And there's also another paper, I haven't clipped the article here, but there's a European one as well, showing very similar results. And so again, as you can see here, we have a reclassification rate that's a little bit higher than their rate. And part of that is likely due, to the fact that we're not only reclassifying based on the updated guidelines, but we're also reclassifying based on the fact that we're finding abnormalities that were not detected by the conventional techniques previously. So, and there's a lot of things that we're also seeing. Nothing in this group here that changed significantly the prognosis in this cohort, but that certainly signal that it's possible, and it's something that we need to look for. And this is the fact that there are structural variants that overlap genes that are normally ascribed to have SNVs in them and have significant outcomes in acute myeloid leukemia due to loss of function, but that could also occur as a result of a disruption by an SV. So, for example, like B-core deletions or RUNX1 deletions, for example, these would be things that could be prognosis altering and not detected by conventional myeloid panels. The other thing that's important here, of course, is the new biallelic TP53 category, where you've got TP53 mutations here detected by our NGS heme, and then in this case we have very clear OGM data showing that the other allele has been affected by a structural variant, usually a deletion that knocks out the other allele of TP53, giving you your biallelic TP53, or in the other case where we have two TP53 mutations with high enough VAF to be considered biallelic. So again, all of this information is, when we put it together, is giving us a lot finer detail that's important for classification. And of course, as I said, there's a 22% change in diagnosis or prognosis as a result of a undetected, previously undetected SV that was detected with OGM. One of the other final points I just wanted to make about this data set was that there's a Ellie Papamanuel paper in 2016 which looked at AML drivers and their percentages in the literature. And there's a bunch of, I put the percentages of those in brackets after the percentage of the actual each of these abnormalities seen in our cohort. And so one of the things I did want to point out is that our percentage is, even in this very small cohort, are actually pretty close to those reported in the Papamanuel paper, which was nice to see, although these ones in orange were actually a little bit higher. This again isn't terribly surprising. These are KMT2A rearrangements, MECOM rearrangements, and NUP98 rearrangements, which probably suggest that in the larger AML cohorts that were published, like the Papamanuel one in 2016, these small cryptic rearrangements that weren't routinely tested for prior to 2015, when the data would have been completed, they're probably under ascertained in those cohorts. The other item that is not actually in the ELM that I put here is the ability of OGM to detect KMT2A partial tandem duplication. So this is just an add-on advantage. It's currently not part of the ELN classification for AML. It's obviously more important in other myeloid diseases like myelodysplastic syndrome from a prognostic perspective. But also the metadata that's being published, or sorry, the meta-analyses, I should say, that have been published in this area suggest that the KMT2A does actually have, PTD does have an adverse prognostic impact. And so it's likely that the PTD may be included in a future version of the classification. And so one of the, some of the interesting things we've seen looking at PTDs, obviously, by OGM, is we can see a variety of sizes and structure of the PTDs. So from 23 kilobases, 32, which is sort of the standard one that we normally see, up to 43 kilobases. And then this paper here off to the right, just to give a couple of examples of the fact that there are actually a variety of different mechanisms of PTD. So, here's your simple PTD, which is, we think, is this 32 kilobase one. You can have double gains of PTD and copy neutral LOH PTD, higher order tandem gain PTD. So there's a whole bunch of mechanisms here that that are potentially at work. And I think there's also the possibility that not all of these PTDs have the same functional outcomes, and that's a question that that's going to have to be dealt with through further research. But it's certainly now with these technologies that we're using, we're getting a lot more detailed information about this, and these are questions that are now tractable that can be answered, which is exciting as well. So just to comment very briefly on the reclassification rates, you can see, just sort of how the different patients were reclassified, what the proportions of reclassification were. There was a relatively low reclassification rate in the favorables, so most favorables remained favorables. A lot of the intermediates remained intermediates, although there's a fairly significant proportion that were classified up to adverse, and a lot of this has to do with the inclusion of the mutations, which is very, very consistent with what we saw in our data, and also, again, very few downgraded from adverse based on the new classification. And of course, there were some ambiguous ELN classifications from the original 2017, and so a lot of these were classified as well in the 2022 classification. So anywhere where the classification has improved, and being able to better classify patients obviously is an improvement. And so I don't need to actually go over this because we've already done that. So that was just basically, I just basically wanted to share that data with everybody, and that's basically it for me, I guess. So I'm gonna stop sharing. Okay, and open it up for questions. Just while we're waiting for questions and comments on that, I just wanted to just point out before you guys start to leave that there is a link in the chat to fill out a survey to request a CPD certificate. So if you are, if you want to, this is an accredited Section 1 activity by the Royal College. So if you want your Section 1 credits, you have to fill out the survey, and then you get a nice certificate that you can put in your CME folder and log it for CME hours. All right. Well, it looked like everybody's tired. No more questions, no more concerns. I appreciate everybody coming out this afternoon to the community of interest, community of practice, and look forward to future meetings. If you have any suggestions for presentations or cases that you'd like to present, you don't have to wait for my email. Please reach out and get in contact with me and let me know so we can help in planning future meetings and making sure we're on target with what people in this group want. The other thing is we focused a lot also in these first meetings on hematologic malignancy related applications. So if anybody has anything that's either constitutional, prenatal, anything like that that you'd like to present, that would be great as well. So anyways, appreciate everybody's time. Don't forget to fill out the survey, and we'll see you again in a couple of months.